Hi, I'm Aurel Enriquez, and this video contains our discussion on Chapter 14 of Mahon's textbook of Diagnostic Microbiology, which talks about the Staphylococcus species. Here is the outline of our discussion. We'll first describe the general characteristics of the genus Staphylococcus, and then describe the virulence factors associated with the Staphylococcus species and their role in pathogenesis. Next, we'll evaluate the catalase and coagulase test for the identification of Staphylococcus aureus, and finally, we'll look into the other coagulase-negative staphylococci. Let's proceed with the clinically significant species. Starting off with the general characteristics of Staphylococcus species, the genus name for Staphylococci was described or was derived, was derived from two Greek words, staphel, which means bunches of grapes, and kokos, which means berries. And this is because of the microscopic appearance after gram staining, as shown here on the picture to the left. They are gram positive, spherical cells or cocci in clusters. So ganun po yung dapat na description natin when we are um, reporting the morphology of bacteria after gram staining. Sasabihin mo muna yung gram staining reaction, tapos sasabihin mo yung shape, and then sasabihin mo yung arrangement. In this case, they are gram positive. What is the shape? Cocci. What is the arrangement? Clusters. Okay, so again, I repeat, no, when properly reporting the morphology of bacteria after gram staining, you should first mention the gram stain reaction, and then its shape, and then its arrangement, if there is any arrangement. So for staphylococci, they are described as gram positive, cocci in clusters. All right. So another general characteristic would include that they are catalase positive, oxidase negative, and facultative anaerobes. Now, if you would remember from previous discussions, when we say facultative anaerobes, they're able to survive with or without oxygen, but they grow better with oxygen, di ba po? And they're capable of surviving exposure to oxygen thanks to the presence of the enzyme catalase, which can decompose hydrogen peroxide. And ayun nga po, no, hydrogen peroxide kasi can be toxic to the bacterial cells. All right. Now, if you would remember from previous discussions, di ba po, part of the killing mechanism of phagocytes is the respiratory burst, which could produce great amounts of hydrogen peroxide. So, ayun po, no, I hope you're able to follow along. Everything's falling into place, di ba? Everything's being connected. Nagagamit na natin ngayon yung mga information na na-discuss natin sa previous chapters. Alright? Next, staphylococci are also non-motile. So, they don't have any form of flagella. They are also non-spore forming and hindi nila kayang mag-produce ng endospores. On blood agar plate, they appear to be small or actually sometimes they range from small to medium eh. Nagre-range po yung size na yun, no? They are small to medium. Sometimes we may be able to observe large colonies. But other descriptions would include that they are creamy or buttery looking, all right? So they may either be white, light gold, or yellow, or gray in color. But as we've discussed previously no, on chapter 8, diba, use of colony morphology, isa sa mga pinaka-iconic na description ng Staphylococcus aureus is that they are capable of producing golden yellow colonies, di ba po? What pigment is responsible for this? Staphyloxanthin. All right? So, ayan po. That's for the description of colonies on the blood agar plate. So, on to these specific species, let's start with Staphylococcus aureus. This is the most virulent species in the genus, and it could cause an infection when it enters a normally sterile site. All right? So, bali harmless naman po most of the time. Ang Staph aureus considered ito as normal flora as long as it stays on the surface of the skin. All right? Kaso lang, if they would be dislodged or moved to sites that are normally sterile, then that's when they may be able to cause an infection. Okay? So marami pa pong iba't ibang virulence factors ang Staph aureus, but its primary virulence factor is the enzyme coagulase or staphylocoagulase. As I've mentioned earlier, they may be halophilic, so we may be able to observe or isolate them using the mannitol salt agar as shown here on the left. Alright, isa-isahin na po natin yung mga virulence factors ng Staph aureus. First is coagulase or staphylocoagulase. The mode of action of coagulase is coagulating the fibrinogen in the patient's plasma. Kapag nangyari po yun on the staphylococcal abscess that were formed during the course of infection, they will form a fibrin layer that can protect the bacteria from phagocytosis. So now, the fibrin layer, no, using the fibrin layer, matatakpan po sila. So, hindi sila mahanap or madedetect ng mga phagocytes. There are two types of coagulase, the bound and free coagulase. The bound or cell-bound coagulase is attached or bound to the cell wall and can clot human, rabbit, and even pig plasma by converting fibrinogen into fibrin. Free coagulase does the same thing, but this can be found extracellularly. Okay, so hindi po katulad ng bound coagulase that is stuck on the surface of the staphylococcal cell wall. Ang free coagulase po, pinoproduce lang ito ng bacteria. Okay, ulitin po natin. When we say cell-bound coagulase, this coagulase molecule is incorporated onto the structure of the bacteria. But when we say free coagulase, this is coagulase that is just released by the bacteria. It's not incorporated onto their cell wall structure. Next is staphylokinase or fibrinolysin. Staphylokinase causes a fibrinolytic activity by dissolving fibrin clots, which is the opposite of what coagulase does, right? So kung magfo-form po kasi ang Staph aureus ng fibrin clot, sila din mismo, they can't easily get out of that clot that they have formed. So hindi sila makakapag-move from one infected cell to another. So after nilang maiwasan yung phagocytosis, kailangan din po nilang makaalis doon by dissolving the clot with staphylokinase. So, staphylokinase helps Staph aureus to cause disseminated infections instead of just being localized. Next is protein A. All right. So protein A, it helps avoid phagocytosis by binding to the antibody that should supposedly be recognized by the phagocytic cell. All right. So it prevents phagocytosis by binding onto the antibody imbis na yung antibody pa yung dumikit sa antigens ng Staphylococcus aureus. Hyaluronidase, otherwise known as the spreading factor, enhances the invasion and survival of bacteria in tissues by breaking down hyaluronic acid that is present on the connective tissues of the patient. This would help the bacteria spread further to other sites. Kaya po siya tinawag na spreading factor. Deoxyribonuclease or DNase can lower the viscosity of exudates by destroying the DNA content of these substances, which would also give Staphylococcus aureus more mobility katulad ng hyaluronidase. So it would facilitate the invasiveness of Staphylococcus aureus. All right, so both hyaluronidase and deoxyribonuclease, it decreases the viscosity of our the tissue fluids, all right? Ang pinagkaiba lang po ay yung target nila. So hyaluronidase would destroy hyaluronic acid, DNase would destroy DNA. So magiging less viscous and more liquidy or more runny yung fluids within our tissues, giving them ayun nga po no, the capability to spread to other places. All right? Kasi po 'di ba, ang Staphylococcus ay walang flagella. All right. So they don't have any appendage that could help them move. All right. So they would have to rely on other mechanisms to move from one place to another dahil nga wala silang flagella. All right. Next is lipase. Lipase is a fat splitting enzyme, and this is essential for bacterial survival in places such as the sebaceous glands. All right. So, di ba po, na-discuss natin sa previous chapters that um, we talked about this no sa composition ng normal flora. Kadalasan po lipids or fatty acids, they may be toxic to some bacteria. All right? Ulitin po natin no, our skin has glands and those glands produce lipids or fatty acids and those fatty acids could be toxic to some bacteria. But for the pathogenic strains of Staphylococcus aureus, they can produce lipase. All right? So when they produce lipase, they're able to survive and colonize areas where humans have sebaceous glands, such as hair follicles. So lipase contributes to the formation of furuncles, carbuncles, and boils. Okay? So mamaya po, pagdating natin sa clinical manifestations, iisa-isahin po natin yung mga diseases na nakalista dito. Okay? So if you notice, no, these three different virulence factors, hyaluronidase, DNase, and lipase, they contribute for the same purpose, which is to help the bacteria spread and infect more sites. Enterotoxins A, B, and D are responsible for cases of food poisoning. All right, so Staphylococcus aureus could also cause food poisoning. Enterotoxins are heat stable, so they are effective for up to 100 degrees Celsius even after 30 minutes of heating. Hindi daw po masisira ang structures at hindi maaapektuhan ang effectivity ng enterotoxins kahit initin pa natin yung food item ng 100 degrees Celsius at kahit hanggang 30 minutes pa. Alright? So, ayun po, no, kahit initin nyo po yung food item ninyo, you may still be able to experience or show symptoms of food poisoning if Staph aureus is the causative agent. Alright? Because the toxin that it produces is heat stable. Alright? So, naintindihan po ba, no? Even though this temperature may be enough to kill some bacteria, it's not enough to inactivate the enterotoxin that was already produced by the bacteria. Enterotoxins are also resistant to gastric enzymes. So, wala lang sa kanila yung hydrochloric acid ng stomach. They are neurotoxins, all right? So their mode of action is to stimulate vomiting. The most common enterotoxin is enterotoxin A, while enterotoxin B is associated in cases of pseudomembranous enterocolitis, which is often found in contaminated milk products. TSST1 or the toxic shock syndrome toxin 1 is a pyogenic exotoxin. So it causes almost all cases of menstruation related toxic shock syndrome. What happens here is that this toxin stimulates the production of large amounts of cytokines, which would lead to the production of symptoms. Okay, so what are cytokines? Now, just to simplify, what are cytokines? These are substances that are secreted by certain cells of the immune system that could affect other cells in the body. So parang sobrang activated ng immune system or immune response na nagiging OA yung reaction ng immune response ng pasyente that it is now starting to affect the other organ systems and functions of the body, and it's actually causing more harm than good. All right. So again, the toxic shock syndrome toxin 1 is capable of triggering the release of large amounts of cytokines, excessive amounts of cytokines. The two toxins here on this slide, I've decided to put them together because they're both responsible for causing skin infections. So first up is PVL or the Panton Valentine leucocidin, which is, I think it's something I've already mentioned in previous discussions. Di ba po? This is a cytolytic toxin that can kill macrophages, monocytes, and neutrophils, which would help the bacteria evade phagocytosis. Plus, this could also cause necrosis of the infected skin tissues and it could also lead to other soft tissue infections. Next is exfoliatin A and B or epidermolytic toxins A and B. These ones are proteases. So when we say proteases, sorry, they could destroy protein structures. All right, again, proteases, are capable of destroying protein structures, right? So these ones, no, specifically, they could destroy the intracellular bridges on the epidermis, which causes excessive sloughing off of the skin at the epidermal layer, causing a burn-like effect. Okay? So, natutuklap po ng sobra at parang nalalapnos po yung balat ng pasyente that are suffering from the scalded skin syndrome because of the epidermolytic toxins. The next virulence factor contributes to antimicrobial resistance. Strains of Staphylococcus aureus that can produce beta lactamase accounts for about 90% or greater than 90% of all isolates that have shown resistance to penicillin and other beta lactam drugs. Okay, so the mode of action of beta lactamase is to break down penicillin, as we can see here in this reaction. Okay, so we start off with penicillin, but after the action of beta lactamase, it gets converted to penicilloic acid. Unfortunately, penicilloic acid no longer has any antimicrobial properties. All right, so again, beta lactamase is a contributor to antimicrobial resistance, specifically for penicillin. Now that we know kung ano-ano yung mga virulence factors ng Staph aureus, let's now look into what clinical manifestations or diseases can they cause. So let's start with cutaneous infections. Folliculitis is the mild inflammation of the hair follicle or sebaceous gland. So in folliculitis, individually infected yung bawat hair follicle nung pasyente. So there could be many hair follicles that are infected at the same time, as we can see here in this first photo, but they are infected separately. Let's compare that with a furuncle. So furuncles are large superficial abscesses that is an extension of folliculitis. So dito po, multiple hair follicles or multiple sebaceous glands in one area that are infected have joined together into one abscess. As we can see here in the second photo, hindi na natin ma-point out yung individually infected hair follicles, unlike here in the first photo. Carbuncles are just multiple furuncles, but the downside here is that this may now progress to a systemic infection. All right. So, systemic infection meaning the patient may now be experiencing fever and chills. So, hindi na po siya simpleng skin infection lang. Okay. But now the patient is starting to show other symptoms. So, this third photo shows us what a carbuncle looks like. Next is impetigo. So impetigo is a superficial cutaneous infection that is common among children, characterized by crusty lesions plus vesicles plus red borders surrounding those lesions. All right. So this thing at the top lip of the patient, no, those are vesicles, and the one at the nose, no, that's a crusty lesion. So notice how red it is and its immediate surrounding. So pag sinabi naman natin crust or crusty, it means may pieces po ng skin na parang tumigas na at pwedeng matuklap. Alright, so again, this is impetigo, which is common among children. Let's now proceed to toxin-induced diseases by Staphylococcus aureus. The scalded skin syndrome, as shown here on the photo to the left, leads to extensive exfoliative dermatitis caused by the virulence factor exfoliatin. So we've talked about this in the previous slides, diba? Exfoliatin or epidermolytic toxins. They're capable of destroying the linkages between the epidermal layer of our skin. So this occurs primarily in newborns and in some children. So as we can see right here, ayun nga po, no, nagtutuklap or parang nalalapnos yung balat ng pasyente. Next is the toxic shock syndrome or TSS. This is a rare but fatal multi-system disease characterized by fever, chills, vomiting, diarrhea, muscle aches, rashes, and could rapidly progress to hypotension and shock. Alright, so nabanggit ko po ito kanina, di ba? The toxin responsible is the pyogenic toxin TSST1, which is capable of triggering the release of excessive amounts of cytokines, which again, cytokines are compounds that are supposed to help us fight infections, di ba po? But again, that's good in moderate amounts. So in excessive amounts, cytokines could also cause more damage or harm to multi-organ systems. Listed here are all the other diseases that could be caused by Staphylococcus aureus, though we will no longer be discussing them one by one since they're not actually unique to Staph aureus. Okay, so marami pong iba pang bacteria na kayang magdulot ng mga sakit na nasa listahan na to. Let's now look into the microscopic and macroscopic morphology of Staphylococcus aureus. As mentioned earlier, sa general characteristics, they are gram-positive cocci that may appear singly or in clusters, as we can see here in this example. All right, so ulitin natin, they are gram-positive cocci in clusters. On culture media, such as the blood agar plate or sheep blood agar, they produce colonies that are yellow, round, smooth, and beta hemolytic, as shown here in this second photo. All right. So notice those cleared out zones of beta hemolysis dun sa close-up shot po dun sa picture. So again, Staphylococcus aureus is beta hemolytic. On mannitol salt agar or MSA, Staphylococcus aureus will produce yellow colonies surrounded by yellow halos, and this is due to the fermentation of mannitol in the culture media. Originally, red po yung color ng mannitol salt agar because it contains phenol red. But as the bacteria ferments the mannitol, it creates an acidic environment, and this changes the original red color into yellow. So pag na-observe po natin no, na ang isang gram-positive cocci in clusters is capable of growing at the mannitol salt agar at nag-produce ng yellow halo sa paligid ng um, colonies nila, this could indicate or presumptively tell us that your isolate may be Staphylococcus aureus. Next is the chrome agar or chromogenic agar for MRSA or methicillin resistant Staphylococcus aureus. So, as we can see right here, no, in this last photo, all right, ang purpose po ng chromogenic agar is for us to know if a bacterial isolate is an MRSA or a methicillin resistant Staphylococcus aureus. So, based on the manufacturer, these chromogenic agars may have different indicators depending on the color of the colonies, all right? Or depending on the color of the colonies that we would be able to observe, no? Doon po natin malalaman kung MRSA ba yung isolate or not. So usually, if they appear as mauve pink colonies, tulad nitong nasa example na to, then it is most likely the isolate is in fact methicillin resistant. So those are the different appearances of Staphylococcus aureus on gram stain and on different culture media. Let's proceed to the other methods of laboratory diagnosis. First, we have catalase and coagulase. So, catalase, as previously discussed, is an enzyme that helps bacteria survive from exposure to hydrogen by converting it to just water and hydrogen, as we can see here in this chemical equation. Now, if you would remember, di ba po, hydrogen peroxide is toxic to a lot of bacteria. The catalase test can be used to differentiate catalase producers from non-catalase producing bacteria like streptococci. So basically, staphylococci are catalase positive and streptococci are catalase negative. So using 3% hydrogen peroxide, the catalase test could be done in a few different ways. First is on a slide. So just take a slide, then place a drop of 3% hydrogen peroxide on a clean glass slide and take an isolated colony from the culture media and then mix that with the drop of H2O2. A positive reaction is indicated by abundant bubble formation, as we can see here in these examples. So, this may also be done on a tube kung hindi naman kayo nagtitipid sa reagent. So just place 1 ml of 3% hydrogen peroxide on a clean glass tube, pick up colonies and then mix and then observe for abundant bubble formation. just like on this picture. Another way of doing the catalase test is by just directly dropping the reagent onto a colony on the agar, just like this photo. So, pagdating nyo po sa internship nyo or sa work nyo, make sure to check with your laboratory staff kung anong method yung sinusundan nila to perform catalase before actually doing anything. Because there are some laboratories out there that don't recommend directly dropping the catalase reagent onto the blood agar plate, diba? Because our RBCs might also contain catalase, which could produce a false positive reaction. All right. So the best way and the most common way that we usually do it is using a slide. Right. So again, wag po palaging tutularan itong nasa third picture. All right. Next is coagulase. So the coagulase test, the reagent for this is rabbit plasma. So for the slide method, the steps are the same as catalase, but with a different reagent. All right. So place 1 drop of the catalase reagent on a, sorry, coagulase na. 1 drop of the coagulase reagent on a clean glass slide, pick up a colony and mix them together. A positive result on the coagulase test is indicated by obvious clot formation, as we can see here in this picture. All right. Here in the negative test result, kung may nakikita man po kayo dyan na granules, that's probably just clumps of bacterial cells from the colony that you got that you were not able to mix very well. Okay? So kailangan apparent and abundant yung amount nung clumping na na-observe natin for us to say that the coagulase result is actually positive. Now, for the tube method, again, same steps, different reagent, and a positive result is indicated by clot formation and cloudiness in the tube, as we can see right here in the bottom right example. Okay, so that's for the tube method. May dalawang klase ng coagulase, di ba? So magkaiba rin po yung dinedetect ng bawat test na to. So the slide method would detect bound coagulase. So pag sinabi ulit natin na bound coagulase, yun yung coagulase that is incorporated on the cell wall structure. Pero kapag sinabi nating free coagulase, those are the coagulase that could be released or secreted by the bacterial cells. And we're able to detect the production of free coagulase using the tube method. Let's proceed to the differential test. So, kanina po, di ba, nabanggit na natin tong isang to, mannitol fermentation. So, mannitol fermentation is done using the mannitol salt agar. This is performed to differentiate pathogenic strains from non-pathogenic strains of the bacteria. So, pathogenic Staphylococcus aureus will produce a positive result with yellow colonies that are surrounded by yellow halos or a yellow discoloration on the culture media. This means that they were able to survive the high salt concentration of the agar, and they were also able to utilize mannitol on the agar as a carbohydrate source. So as I've mentioned earlier, MSA uses phenol red as a pH indicator. So if the bacteria was able to produce an acidic environment after mannitol fermentation, the color changes from red to yellow. So notice, 'di ba po, dito sa areas nung um culture media na to na walang tumubo na colonies, no, it's showing us na color red pa rin because again, the original color of the mannitol salt agar is red. So again, MSA is used to differentiate pathogenic strains from the non-pathogenic strains of Staphylococcus aureus. Because again, kung pathogenic ang strain ng Staph aureus na na-isolate ninyo, it could ferment mannitol and it could survive high salt concentration. The deoxyribonuclease test or DNase test is used to check if the pathogenic isolate can produce the enzyme deoxyribonuclease. Marami po kasing strains of Staph aureus are pathogenic, but not all of them can produce DNase, which is why we need to do this test. The culture medium used is the methyl green agar, but for some manufacturers, they could also use toluidine blue. But anyway, in this example, we have the methyl green agar, and here are its optimum incubation conditions. The isolate must be kept at 35 to 37 degrees Celsius for 18 to 24 hours before reading the result. The positive result is indicated by a formation of a clear zone around the bacterial growth. So as we can see here in this photo to the left, they tested three different bacteria. We have Staphylococcus aureus, next is Serratia marcescens, and next is the Enterobacter species. So positive results were produced by Staphylococcus aureus and also Serratia. Alright? So kaya nakikita nyo po na wala yung color sa paligid nila. Meanwhile, Enterobacter tested negative because although it was able to grow, it did not clear the methyl green in its immediate surrounding. Now, what is the principle behind this test? All right. Ang methyl green agar po, it contains DNA molecules that are complexed or attached onto the methyl green dye. All right. Ulitin po natin, no. The DNA molecules are bound together with the methyl green dye. Now, if the bacteria is capable of breaking down the DNA molecules, this also destroys the methyl green and DNA complex. So, pinaghihiwalay po ng DNAs yung binding ng methyl green at saka DNA, which would result in the clearing out of the area where the DNAs was able to be or was able to act upon. Okay? So again, that's the principle for the DNA test. The culture media that we're using is the methyl green agar. The methyl green agar contains the methyl green dye, which is complexed or bound to DNA molecules. If the organism produces DNAs, sisirain niya yung structure ng DNA at wala na pong kakapitan yung methyl green agar dye or methyl green dye. So we won't be able to observe them anymore. Kaya may complete clearing. No? So again, a positive result in the DNA test is a clear zone. Next is the PYR or the pyrrolidonyl acrylamidase test. It detects the presence of PYR enzyme which could be produced by Staphylococcus aureus. So pwede natin itong gamitan to differentiate Staph aureus and Staphylococcus lugdunensis. Alright? So a positive result is indicated by the formation of a cherry red color after the addition of the detector reagent. Alright? So again, kung i-differentiate natin ang Staph aureus and Staphylococcus lugdunensis, pwede nating gamitan ng PYR. Staph aureus is negative, alright? Staph aureus is negative, while Staphylococcus lugdunensis would be positive for PYR. Next is the chromogenic test for MRSA using the chrome agar. MRSA is a type of Staph aureus that is resistant to antibiotics such as methicillin or oxacillin. The mode of transmission is through close contact with an infected carrier. There are three types of MRSA. They may be hospital acquired, community acquired, or healthcare associated community onset. So methicillin resistance could be acquired after a prolonged stay in the hospital or after extended antibacterial therapy. And with the MRSA, this may also be a nosocomial or hospital acquired pathogen. All right. So this is common among ICU and burn unit patients because they're the ones na matagal po yung confinement, di ba po? So medyo mas matagal yung incubation period if we're trying to do a chromogenic test. We should keep the isolates for 24 to 48 hours before reporting the result. So as shown here in these pictures, depende po sa manufacturer yung color change that could indicate if the isolate is MRSA or not. So figure A is an MRSA isolate from one patient plated on the high chrome agar. All right, yun yung parang brand name niya, the high chrome agar. Meanwhile, yung figure B naman po is also another MRSA isolate, but this time it was plated on the chrome agar. Okay, so ayun po no, different brands or different manufacturers could have different color indicators that could tell us na yung isolate na nag-grow natin is a methicillin-resistant Staphylococcus aureus. Let's now proceed with the clinically significant coagulase-negative staphylococci. Let's now just quickly discuss all the other Staphylococcus species that are referred to as cons or coagulase-negative Staphylococci. So kung kanina po, we learned that Staphylococcus aureus is both catalase and coagulase positive. Ang cons naman po, well, as the name implies, no, they are catalase positive but coagulase negative. So as you can see right here, first up is Staphylococcus epidermidis. This one is also part of the normal flora, but it is usually seen as a contaminant of medical instruments, catheters, prosthetic heart valves, and other stuff like that. The disease associated is healthcare acquired UTI, but if we were to make use of the mannitol salt agar, Staphylococcus epidermidis will not grow. Okay, so Staphylococcus epidermidis is negative for growth if you would use the mannitol salt agar. Next is Staphylococcus saprophyticus. This causes a disease that is common among young women or sexually active women, which is a form of UTI. Okay, ulitin po natin. Staphylococcus saprophyticus is a common causative agent of UTI among young women. Complications could also lead to cystitis. Now, on BAP or the blood agar plate, they usually produce white and non-hemolytic colonies. Okay, so yun po, no, paano natin ma-differentiate naman yung staph aureus versus staph saprophyticus? Again, on BAP, staph aureus is golden yellow colonies that are beta hemolytic. Pero kung staph saprophyticus po yung na-isolate nyo, they are white and non-hemolytic. Next is Staphylococcus lugdunensis, which is identified as coagulase negative via the tube method. Now, if you would remember in our previous slides, diba, we talked about the coagulase test, and there are two ways to perform it. So, the slide method and tube method. Sinasabi po dito that Staphylococcus lugdunensis may test negative for coagulase kung tube method yung gagawin. But they may appear coagulase positive using the slide method. Staphylococcus lugdunensis, a lot of the strains for this one is also known to carry the MEC-A gene. The MEC-A gene codes for oxacillin resistance. All right. Next is Staphylococcus hemolyticus. This one is being recognized as an emerging nosocomial pathogen, and they tend to develop vancomycin resistant. But again, do take note, emerging pathogen pa lang naman ito, so hindi pa siya commonly isolated or commonly recognized in the laboratory. That ends our discussion on chapter 14 of Mahon's textbook of Diagnostic Microbiology, which talks about the Staphylococcus species. This has been Aurel Enriquez. Thank you so much for watching.